You know, when we think about a medical diagnosis, there's usually this expectation of, like, total precision.
Right, yeah, like it should be engineering.
Exactly. If you break your arm, the X-ray shows that jagged white line. The doctor just points at it and says, yep, there it is.
It feels very binary, like it's broken or it's not broken, it's clean.
Right. But the second you step into the world of cancer pathology, that comforting precision kind of, well, it vanishes. We're looking at a diagnostic landscape that is, honestly, incredibly murky.
Oh, it is the absolute definition of diagnostic muddy waters. And the stakes for clearing up those waters, I mean, they couldn't be higher.
Which is exactly why this deep dive is custom tailored just for you. Today, our mission is to explore a truly groundbreaking 2026 paper. It was published in the journal Cell by Liv and colleagues out of Stanford University.
Such an amazing paper.
It really is. They've essentially figured out a way to completely change how we look at cancer biopsies using an AI platform called Canvas.
Yeah, Canvas.
And it acts like this universal translator, taking simple, really cheap pathology slides and revealing the hidden, complex ecosystems of tumors. And it does this to predict two massive things: if a patient will survive and if they'll respond to immunotherapy.
And the sheer scale of the problem they're trying to solve here is pretty staggering. To really grasp it, I think it helps to picture a clinical scenario based directly on this research.
Oh, yeah, let's do that.
So imagine two patients, right? Both are just diagnosed with non-small cell lung cancer or NSCLC.
Okay, so both patients go in and they both undergo a standard biopsy. The lab takes that little piece of tissue, they slice it incredibly thin, and put it on a glass slide.
Yep, the standard procedure.
Right. And they use a standard stain called H&E hematoxylin and eosin. The hematoxylin stains the cell nuclei purple, and the eosin stains the rest of the cell pink.
It's super cheap. It's been around for, what, over a century?
Literally over 100 years. It's what every hospital in the world uses.
Exactly. And when the pathologist looks at those standard H&E slides under a microscope, both patients look broadly similar. I mean, the cancer looks the same.
Right.
So both are prescribed the exact same treatment immune checkpoint blockade therapy, which is, you know, a drug designed to essentially take the brakes off the patient's own immune system so it can hunt down and fight the cancer.
But then the outcomes completely diverge. One patient experiences long-term survival, their tumor shrinks, and the drug works absolute miracles.
Yeah.
But the other patient, their tumor grows aggressively, the drug fails completely, and their disease progresses. It leaves you wondering, like, why? They have the same disease, the same visible biopsy results, the exact same treatment.
Well, the answer lies in the limitations of that 100-year-old staining technique. Standard H&E staining is fundamentally like looking at a satellite image of a city.
Okay, a satellite image.
Yeah. You can see the concrete structures, you can see the broad outlines of the buildings and the major highways, but you have absolutely no idea what is actually happening down inside them.
Right, you just see the roofs.
Exactly. You don't know who is talking to whom, where the traffic jams are, or, importantly, where the bad actors are hiding out.
Right. And today we are going to learn how to get the street-level view of that city using AI. But to understand how this algorithm eventually learns to read that blurry satellite map, we first have to understand how scientists painstakingly map the city by hand.
Right, which brings us to a concept called the tumor microenvironment and a piece of technology called Codex.
Yes. So let's get into the tumor microenvironment, or TME. We really need to define this exhaustively here.
Definitely, because for a very long time, medical science thought of a tumor as just a rogue lump of mutating cancer cells, just this monolithic bad guy.
Just a ball of bad cells.
Right, but it is not. It's a bustling, highly organized ecosystem.
Almost like a twisted version of a thriving metropolis.
Precisely. Alongside the cancer cells, you have this massive population of immune cells. You have T cells, which act like the foot soldiers actively trying to attack the tumor.
We're good guys.
Usually, yeah. You have B cells, which are producing antibodies. You have macrophages, which usually act like the garbage trucks of the immune system, cleaning up debris.
But wait, can't those be hijacked?
Yes, exactly. They can actually be hijacked by the tumor to help it instead. And then you have stromal cells, like fibroblasts, which build structural scaffolding. And endothelial cells constantly building new blood vessels to feed the growing tumor.
So it's an incredibly crowded, chaotic city. And to actually see all of these different players, the researchers used a spatial proteomics technology called Codex.
Right, which stands for co-detection by indexing.
Yeah. And if the standard purple and pink H&E stain is like a blurry black and white security camera, Codex is a 41-channel, 4K ultra-high-definition camera.
That's a great way to put it.
It's capable of highlighting exactly 41 different functional proteins on every single microscopic cell in a slice of tissue.
And the researchers didn't just use this on a small sample size either. They performed this mapping across over 18 million individual cells from 457 patients with non-small cell lung cancer. 18 million cells. That's wild.
It's a massive data set.
Yeah.
They used those 41 distinct markers to pinpoint exactly who was who. For example, they used a marker called CD8 to tag the cytotoxic T cells, the assassins.
Okay, CD8 for the assassins.
Right. They used Chi67 to tag cells that were actively dividing. And they looked for PD-L1, which is a specific protein that tumors use as a cloaking device to hide from the immune system.
And when they finally had this Ultra HD map of those 18 million cells, they noticed a massive difference between the two main types of lung cancer, lung adenocarcinoma, or LUAD, and lung squamous cell carcinoma, LUSC.
The immune signatures were completely distinct. LUSC showed a much higher overall infiltration of immune cells, but they were mostly what we call myeloid cells.
Which are what, exactly?
Think of your innate immune cells, like neutrophils and M2 macrophages. And those M2 macrophages are particularly problematic because they actually suppress the body's immune response.
Oh, wow.
Yeah, they actively help the tumor heal and grow. While LU8, on the other hand, was enriched for T helper cells, which are part of the adaptive immune system that is actively trying to orchestrate an attack against the cancer.
Exactly.
So just making sure I'm following. We used to think of tumors as just a growing ball of identical bad cells.
Yeah.
But they're actually functioning cities. And this Codex tech gives us the ultimate high-def map of exactly who lives where down to the single cell.
Spot on.
But wait. Profiling 41 distinct proteins on 18 million individual cells across hundreds of patients sounds incredibly expensive, like technically exhausting, and honestly just impossible for a normal hospital to do.
You are entirely correct. I mean, Codex is a phenomenal discovery tool for scientists in a lab, but it requires highly specialized equipment, wildly expensive chemical reagents, massive computing power. It is a major bottleneck in medicine. We just can't do it for everyday patients.
Right, which is exactly the bottleneck the Stanford researchers set out to solve.
Exactly.
Okay, before we move on to how they solved that, let's do a quick 5-minute recap for you, the listener, to lock this in.
Sounds good.
First bullet point. The tumor microenvironment, or TME, is a complex ecosystem. It's not just a clump of cancer cells. It's got immune cells, blood vessels, and scaffolding.
Right. Second bullet point Codex is this Ultra HD tech that uses 41 markers to map this ecosystem at a single cell resolution.
And third bullet, the researchers found that different lung cancers like LUAD and LUSC have completely distinct immune cell populations.
Exactly.
So here is an interactive question for you to mull over. Think about a map of your own neighborhood. If you only looked at the concrete structures, what vital information about the community and how it functions would you be missing?
That's the exact problem with standard biopsies.
Right. Okay, so getting back to it. Before you can solve how to make the map cheaper, you have to actually figure out what the incredibly expensive map is telling you.
Yeah, because even with 18 million perfectly labeled cells, a list of cells isn't a map.
Right, you have to know who is living next to whom.
Exactly. To figure out the spatial relationships, the researchers used an algorithmic approach, specifically something called spatial LDA. They literally draw a microscopic circle, a 40 micrometer radius around every single cell.
So just a tiny circle around each cell.
Yep. And the algorithm just takes attendance of exactly which cells are inside that little circle.
And by doing this millions and millions of times, they discovered that cells in a tumor don't just mix randomly. They organize into 10 distinct recurring patterns.
Right. The paper called them cellular neighborhoods, or CNs.
And honestly, looking at how they behave, it feels a bit like an old Western, the good, the bad, and the fibrotic.
I love that framing. Let's start with the bad.
Okay, give us the bad.
One neighborhood, labeled CN01, is the tumor core. It's just packed with highly proliferative cancer cells rapidly dividing.
Sounds bad.
Very bad. Then there is CN06. This is a neutrophil-rich zone. It is highly hypoxic, meaning it's starved of oxygen, and it's fiercely immunosuppressive.
So if a patient's tumor has a lot of CN06 neighborhoods, that tumor is actively fighting off the body's immune system.
Exactly. Now let's talk about the good.
Okay.
CN03 is the B-cell niche. where those antibody-producing cells hang out. And CN08 is the T-cell compartment, which is absolutely full of those cytotoxic CD8-positive cells.
The assassins.
Yes, the assassins of the immune system. These are the exact cells you desperately want hunting the cancer.
Finally, we have the fibrotic. CN04 is a stromal fibrotic domain. It is packed with CHAS, which stands for cancer-associated fibroblasts.
Right. Now, in a healthy body, fibroblasts help heal wounds by laying down collagen and scar tissue.
But the tumor corrupts them.
It completely hijacks them. It forces them to build a literal physical wall around the cancer.
So we have these distinct neighborhoods, but who is talking to whom? Like, how do they interact on the borders?
Yeah.
The researchers used neural networks, specifically graph neural networks, to look for what they call triplet motifs.
Basically, 3-cell interactions.
Because it turns out 3-cell interactions dictate life and death in this microenvironment.
They really do. A protective motif might look like a cytotoxic T cell standing right next to a tumor cell, with a T helper cell right behind it, cheering it on and keeping it active.
In that scenario, that tumor cell is likely going to be destroyed.
Exactly. But a dangerous motif is when you have an M2 macrophage next to a tumor cell next to a CF.
The corrupted macrophage tells the immune system to calm down, and the CAF is busy building that wall.
Right. And speaking of that wall, we really need to talk about the barrier score because this felt like a massive breakthrough in the paper.
It truly is. The researchers found a way to mathematically quantify spatial insulation. They calculated the shortest physical path that an immune cell must travel to reach a tumor cell.
And if one of those cancer-associated fibroblasts, the CIF, is blocking the path, the barrier score goes up.
The CFs are literally acting like a castle wall, physically preventing the immune system's assassins from reaching the king.
And the clinical data from the patients back this up perfectly. A high barrier score, specifically in early-stage NSCLC, predicted a much worse progression-free survival.
The cancer came back much faster, simply because the immune system was physically locked out of the castle.
So now the scientists have this perfect high-resolution map. They know the 10 neighborhoods, they know the good, the bad, the fibrotic.
And they know exactly how these fibroblasts build physical barriers that dictate whether a patient lives or dies.
Which brings us to our second recap.
Perfect timing. Okay, bullet 1 cells organize into 10 distinct cellular neighborhoods, or CNs, spanning from immune-active to immunosuppressive.
Bullet 2 triplet motifs reveal that the exact spatial positioning of 3 adjacent cells dictates survival.
And bullet 3, the barrier score, proves that fibroblasts can form a physical wall, preventing immune cells from attacking the tumor.
So here's a question for you. If you were designing a new cancer drug, would you target the tumor cell directly, or would you first try to design a drug to tear down that CIF wall?
That's such a crucial question. Because if the wall is there, it doesn't matter how good your other drug is.
Exactly.
Okay, so if the fibroblasts are building a literal castle wall to lock out the immune system, the obvious question for a doctor is, how do we find out which patients have these walls without spending millions of dollars on a Codex camera?
Right. How do we get these incredible street-level insights for everyone?
Using only that cheap $2 purple and pink H&E slide we talked about earlier.
Enter Canvas.
Canvas. That stands for Cellular Architecture and Neighborhood-Informed Virtual AI-Driven Spatial Profiling.
It's a mouthful.
It is, but it's the platform they built to bridge the gap between the incredibly expensive map and the incredibly cheap satellite image.
The mechanics of how they train this AI are just brilliant. Step 1, they took a highly detailed 41-channel Codex image and a standard cheap H&E image of the exact same tissue slice.
The exact same piece of tissue.
Yeah. And step 2 is where the magic happens. They had to co-register them at the absolute single-cell level.
Wait, how do you even do that?
To do this, they used affine transformation matrices. It's a mathematical formula that perfectly shifts, rotates, and scales one map until every single microscopic street corner aligns flawlessly with the other.
So they laid the millions of cells from the expensive Codex image perfectly on top of the purple and pink cells of the H&E image.
Exactly. And once they were perfectly aligned, step 3 was bringing in a vision language foundation model called MUSK.
Now, this AI model had already been pre-trained on 50 million pathology images. It deeply understands what human tissue looks like.
Right. They essentially told the AI, look at the simple H&E image and predict which of the 10 complex codex neighborhoods is present.
Just by looking at the shapes, the folds, and the dyes.
Yes, and the results were staggering. It worked with an accuracy exceeding 0.83.
Which is wild. The AI is basically looking at the structural footprints, or like the ghosts that these complex proteins leave behind on a standard tissue slide.
That is exactly what's happening. Even though the simple H&E stain doesn't color code the specific proteins, the presence of those proteins naturally changes the morphology of the cell.
The morphology, like its physical shape.
Yeah, the physical shape, how it clusters with its neighbors, the physical tension of the cell membrane. The human eye just sees a sea of pink and purple, but the AI can see those microscopic morphological ghosts.
And because Canvas only needs a basic H&E slide to work, they didn't have to stop at the 457 patients they mapped by hand.
No, they unleashed this AI on over 5,000 patients across 9 different cancer types using huge public databases like the TCGA.
And by applying this AI at a massive population scale, they discovered that tumors generally fall into one of 4 macroecotypes, labeled CI through CIV.
And these ecotypes weren't just random shapes, they were directly linked to very specific underlying genetic mutations.
Like, for example, CI is the myeloid-inflamed ecotype. It has lots of innate immune cells and lots of inflammation.
And the AI found that this specific visual ecotype is heavily linked to BCL2 and BRF mutations.
Which are genes that promote rapid cellular survival and division.
Right. Then you have CIV, which is the immune-active ecotype. This one is dense with lymphocytes like T cells and B cells.
And molecularly, CIV is directly linked to the amplification of HIF-1A and PD-L1.
Because it has so many immune cells already present, but they are just being suppressed by that PD-L1 cloaking device, this specific ecotype is absolutely ripe for immunotherapy.
It is genuinely blowing my mind that an algorithm can look at a 100-year-old staining technique, observe how the cells are squished together, and accurately tell you the underlying genetic mutations of a tumor.
It's revolutionary. Let's do another quick recap here.
You got it. Bullet 1 Canvas is an AI platform that predicts complex molecular neighborhoods from standard, cheap H&E slides.
Bullet 2, the AI learns the subtle morphological footprints left by proteins.
And Bullet 3, applying Canvas at a population scale, revealed 4 universal tumor ecotypes linked to specific genetic mutations.
So think about this question. Can you think of other areas in science, or even your own life, where AI could translate a simple, cheap observation into a highly complex, expensive insight?
That's a profound thought, but it makes me wonder about the immediate clinical stakes. We talked earlier about immune checkpoint blockade, or ICB, and how it cures some people but completely fails for others.
Yeah.
Why are our current tests for who gets the drug so bad? Like, why doesn't it work for everyone?
Because the current biomarkers we use, like measuring overall PD-L1 levels or tumor mutational burden, are fundamentally flawed. They rely on bulk measurements.
Bulk measurements.
Yeah. To use an analogy, traditional testing throws the entire tumor into a blender and measures the resulting smoothie.
Okay, that makes sense.
It completely ignores the spatial organization we've been talking about. Canvas, on the other hand, doesn't blend the smoothie, it maps the forest.
To map that forest, Canvas extracted 262 distinct spatial features from the routine slides of patients who had undergone immunotherapy.
They weren't just looking at what cells were there. They were looking at the complex spatial math of how those cells were arranged.
For instance, they measured something called dispersion, like using Ripley's K, right?
Yes. This simply asks, are the cellular neighborhoods tightly huddled together in dense clumps, or are they spread out evenly across the tissue, wandering alone?
They also measured something called spatial transition entropy.
Which sounds intimidating.
It really does. But it just measures the borders between neighborhoods. Think of it like looking at a border between 2 neighborhoods in a city. Is it a clean, straight highway where one side is strictly residential and the other is commercial?
Or is it highly chaotic, with factories and houses deeply intermixed and tangled together?
Right. That level of chaos is exactly what the algorithm is measuring.
Now, 262 mathematical features is way too many for a doctor to use practically. So the researchers used powerful algorithms like lasso-penalized Cox regression and random survival forests to look at hundreds of variables and ruthlessly strip away the noise.
They pared it down to the absolute most predictive core. They whittled those 262 features down to a concise 13-feature spatial signature.
And the evidence for this is incredibly solid. They didn't just test this on one small group. They built the model in a discovery cohort of 149 patients.
And then completely validated it in an entirely independent group of 40 patients from the Cancer Moonshot Biobank.
And the findings were undeniable. The patients who responded well to immunotherapy, the survivors, had a high degree of spatial localization of two specific neighborhoods, HO3 and HO8.
If you remember from our Western analogy, that's the B-cell niche and the T-cell compartment. They were physically working together.
Yes, forming what are called tertiary lymphoid structures.
Wait, what are those?
These are basically pop-up immune boot camps right inside the tumor.
Oh, wow. So the immune system is actively setting up base camps.
Exactly. In stark contrast, the non-responders, the patients where the drug failed to work at all, had a high interaction between the HO4 and HO9 fibrotic domains.
That indicates deep fibrotic suppression. The castle walls were simply too thick for the drugs to help the immune system break through.
But the true breakthrough of this entire paper, the moment that changes clinical practice, is that this 13-feature signature accurately predicted who would survive, even in patients whose tumors were technically PD-L1 negative.
Which is huge. Traditionally, if a patient's tumor tests negative for PD-L1 using the old smoothie method, a doctor might deny them immune checkpoint blockade therapy.
Right, assuming it just won't work.
But Canvas was able to look at the spatial math of the cheap slide and say, no, wait, the microscopic architecture here says this patient is actually a responder.
Patients who might have been denied life-saving immunotherapy based on traditional tests could be identified as responders by this AI and ultimately saved.
That's our final recap moment.
Let's do it.
Bullet 1 Canvas extracts complex spatial math, like transition entropy, from simple tissue slides.
Bullet 2, a 13-feature signature, reliably predicts who will respond to immunotherapy.
And Bullet 3, the AI outperformed traditional clinical biomarkers, finding hope even for PD-L1 negative patients.
And the final interactive question for you, what does it mean for the future of human health when an algorithm can see a clear path to survival that the human eye completely misses?
It's profound. When you pull all of this together, it feels like we are fundamentally shifting how we view disease as a whole. We are moving away from treating a tumor as a blender full of bad cells and moving toward understanding spatial ecology.
Right, mapping the forest.
Exactly. And we're unlocking this entirely through a foundation model that democratizes what used to be a multi-million-dollar insight. It's giving that power to literally any hospital with a basic microscope, a glass slide, and an internet connection.
It's an incredible democratization of science. And I want to leave you with a final paradigm shift to think about. If an AI can predict the exact local architecture of a tumor from a basic stain, the next step isn't just predicting if a single drug will work.
Right.
The next era of medicine is designing spatial therapies.
Spatial therapies.
Yeah.
Like treating the geography of the tumor.
Precisely. Imagine a treatment regimen based entirely on the canvas map. We know the CCAFs have built a physical wall, right?
Right, the castle wall.
So drug A is given on Monday specifically to dissolve that fibrotic barrier and lower the barrier score. Then drug B is given on Wednesday to send the T cells rushing through the newly created breach directly into the tumor core.
So we aren't just poisoning the tumor with broad-stroke chemotherapy anymore?
No, we are running a targeted tactical siege based on spatial intelligence.
That is absolutely wild to think about. It really makes you look at the world differently. Next time you look at a simple image, a basic spreadsheet, or a routine data set at your job, ask yourself, what hidden dimensions is your brain missing that a foundation model might clearly see?
Are we all just scratching the surface of the physical world?
Just staring at the jagged white line on the X-ray, completely blind to the bustling microscopic universe operating right underneath it.
The muddy waters are finally starting to clear.
They really are. Thank you for joining us on this custom-tailored deep dive. Keep looking closer, and we'll see you next time.